Hi everyone, this is Anudh Mishra from AIIMS Bhubaneswar. Today we are going to solve some questions from the chapter Molecular Basis of Inheritance. So this chapter is a part of your class 12th this thing biology, and it's very important because many students find a lot of difficulties in this chapter. They find it to be difficult as a chapter, but if you know the concept, this chapter is not that difficult. You will be able to solve most of the questions. So, first, before starting, I will like to start with the definition of this chapter, what this chapter means. Okay, molecular basis of inheritance. So, first, take a look at this word inheritance. Okay, what is inheritance? So, in simple terms, inheritance is what you pass on to the next generation. So, for example, you have inherited something from your parents. Your parents have inherited some things from their parents, and their parents have inherited things from their parents. So, every organism that reproduces, whether sexually or asexually, they pass down some characters. Okay. What do they pass? They pass characters. These characters are inherited by their offsprings. Okay. So, what governs that these characters are passed? How is it regulated? Because, for your information, if this is the parent and it has 100 characters, this is the other parent, and this parent has, for example, 50 characters. Excuse me. Their offspring, if you cross them, they will have an offspring. This offspring will have, if you are expecting that this offspring is going to have 150 characters, you are wrong. This will have some characters from the father and some characters from the mother. Now, this could have 25 characters from the mother and 70 characters from the father, which makes it 95 characters from the parents, or it may be any variable number. Now, this arrow, what governs how many characters are inherited or what kind of characters are inherited, that is what this chapter explains us. This chapter tells us what governs these inheritance laws. How can you predict what kind of characters the child is going to inherit, and how everything is passed on from one generation to the other? The simple molecules involved in this, how do they interact with each other? Okay, molecular molecular basis of inheritance. So definitely it is going to involve molecules. What molecules are involved in inheritance? First, try to think. What molecules do you think are involved in the inheritance? Yeah, yeah, you're correct. Molecules like DNA, molecules like RNA are involved in inheritance. Okay. So they will be the prime focus in this chapter. DNA structure, RNA structure, how DNA is formed, how RNA is formed, what happens to DNA, how DNA works, what happens to RNA, how RNA works. All of these simple things we are going to see in this chapter. And if you have read it from the NCERT, you know that there are a lot of variations, a lot of possibilities, and the functions of the basic summary of all of this is the central dogma. Okay, the central dogma, which says that DNA is converted to RNA. This process is called transcription. RNA is translated to proteins, and DNA itself replicates to form new DNA. This is called the central dogma of biology. Okay, this was uh, first brought into the table by the famous scientists Watson and Crick when they described the structure of DNA, double helical structure they described. Along with that, they also told us how DNA replicates, how RNA is formed, and this is how everything is passed down from generations to generations. Okay. Okay. So we'll move on to the first question now. Express sequence tags are what? So see, expressed sequence tags. I just gave you this an overview of the central dogma. I will draw it again. DNA is transcripted to RNA. RNA is translated to proteins, and DNA itself replicates to form DNA. Now, suppose this is the DNA. Okay. Suppose this is the DNA. I will extend the length a little bit. DNA contains genes. This is gene A. This might be gene B. This might be gene C. So, even genes contain portions. So, all part of DNA, the whole DNA itself is not expressed as RNA. Okay. All the genes are not expressed as RNA. Okay. Only some genes are able to form the RNA. Only some genes. Then you will say, what are the other genes doing? The other genes are controlling. They're controlling how the expressing genes are behaving. How much of these genes are going to be expressed into RNA. Okay. How much, the frequency, all these things are controlled by these other genes. Okay. So these genes which are getting expressed, gene A, gene C, for example, they are what? They are called expressed sequence tags. Okay. So expressed sequence tags are DNA sequences or genes that are expressed as mRNA for protein synthesis. They are used in the Genome Project. Express sequence tags are small pieces of DNA sequence, usually 200 to 500 nucleotides long, that are generated by sequencing either one or both ends of the express gene. First, each gene must be converted into messenger RNA that serves as a template for protein synthesis. So, what you need to remember over here is basically these genes are the ones that are expressed as mRNA. And this term was basically used for the first time during the Human Genome Project when the whole DNA was being mapped. Okay. During that time, this term first came into play. Okay. Okay. We'll move to the next question. Basis of DNA fingerprinting is. Now, you know what fingerprints are used for, right? Fingerprints are used for identifying the person because everyone has their own unique fingerprint, and no two fingerprints can be alike. Similarly, when you use DNA to identify the person or to establish relations between two persons, it is called DNA fingerprinting. Okay. So, if, for example, you found a DNA at a crime scene. Okay. Now you want to know whether person one or person two, which of them is actually involved in, or which person's DNA is actually, which person's DNA is actually so which person's DNA was actually present at the scene you want to find out. Okay. How will you find out? You will find it out by running a DNA fingerprint. Okay. What will you do is, if this is the DNA, the whole length of DNA, your concern is that the whole DNA is composed of nucleotides. Okay. Nucleotides are this single bases. These have of four types: adenine, guanine, cytosine, thymine. Okay. So you have a sequence of DNA. It is like A, G, C, T, T, A, T, C, G, G, etc., etc. Now, the whole DNA has some parts which start repeating itself. Okay, these parts which start to repeat itself. So, between there are some parts which are actually repeats. For example, A, G, G, C, T is repeated 1,000 times. And this part is called RFLP, restriction fragment length polymorphism. So this RFLP is different for different individuals. It will be different in me, it will be different in you, it will be different in your parents, it will be different between my parents. Okay. So this is what is the basis of DNA fingerprinting. Okay. So DNA fingerprinting is a technique used to identify the person on the basis of his DNA, which is specific for every individual. The technique for the first time was developed by Alec Jeffreys and his colleagues at Leicester University in UK. DNA of an individual carries specific genes, specific sequences of nucleotides, which are repeated many times through the length of the DNA. This is called restricted fragment length polymorphism. Okay. Next, non-membranous nucleoplasmic structures in nucleus are the site for active synthesis of non-membranous nucleoplasmic structures are the site for the active synthesis of what? First, it says nucleoplasm, which means it's in the nucleus. Now, if it's in the nucleus, you know what all structures are there in nucleus. There is no membrane-bound structure in the nucleus. Only one additional structure is there inside the nucleus, that is nucleolus. And that synthesizes what? We all know, synthesizes rRNA. Yes, you are correct. So we'll move on to the next question. Which of the following conditions will there be no change in the reading frame of following mRNA? Now the frame of the mRNA, you read it 3 nucleotides at a time. Okay. So if you insert or delete, you will definitely change the reading frame of your DNA. So option number A and C are not viable. Like they are not correct because you are inserting and deleting stuff over there. Now let's see what all options are left with us after option number A and B. Insertion of A and G at 4th and 5th position respectively. Do you think this will give you the same sequence? So if you just insert or delete any 2 nucleotides at 4th and 5th position, then again you will change the reading frame. But if you delete GGU, if you delete GGU from the 7th, 8th, and 9th position, that will leave you with no change in the frame. So there will be no change in the reading frame of mRNA by deletion of GGU from 7th, 8th, and 9th position. So your answer is D. So whenever you change any multiple of 3, okay, in multiples of 3, if you start changing, you will not change the reading frame because codons are read in a multiple of 3. All of the following are part of one operon except. So an operator, structural genes, a promoter, and enhancer. So if you form the structure of an operon, you will have what? You will have an operator or an operon. You will have the structural gene. You will have the promoter from where it is going to start. But the enhancer is what can be outside of the operon. Enhancer can be outside the operon. So option number D is, it can be your answer. Okay. So the answer will be option number D. Okay. Types of RNA polymerase is required in nucleus for RNA synthesis. So this question is a bit incomplete. First, okay, yeah. So they have asked in nucleus. That gives you a hint that this is a eukaryote, possibly human. So this is not a bacterial RNA synthesis that we are trying to elicit. So in eukaryotes, you know that there are 3 types of RNA polymerase: RNA polymerase 1, RNA polymerase 2, RNA polymerase 3. So the answer will be C. Right? So the answer is C. Eukaryotes have 3 types of RNA polymerases. RNA polymerase 1, it is found in nucleus and is responsible for these rRNA transcriptions. Okay, synthesis. RNA polymerase 2 is found in the nucleoplasm, again in the nucleus, and is responsible for mRNA synthesis. So this is the main RNA polymerase which forms the mRNA, which goes on to form the protein. RNA polymerase 3 will form only 2 special rRNAs, tRNA and 5S rRNA. So you have 3 types of RNA polymerases. Done? Okay. We'll move to the next question. Gene and cistron words are sometimes used synonymously because. Now, gene and cistron. So this is a length of a DNA. It will have many genes. Genes are what? Sequences that have a function. What is a cistron? Cistron is a structure composed of many genes. So you can have a gene over here, a gene over here, a gene over here, some gene in between. All of these might form a cistron. Okay. So cistron is that segment of DNA which specifies synthesis of a polypeptide. Okay. Now DNA, I told you, the whole length of DNA everywhere you will not find genes forming mRNAs, which will go on to form proteins. Proteins are what polypeptide means. So polypeptide is they are talking about protein only. Only specific places will form the mRNA. And these specific segments of DNA are called what? Cistrons. Understood? Now mutation generally produces. So they have asked generally. So first we'll understand what is mutation. In simple terms, I told you the central dogma. This is DNA. And this DNA is going to replicate and form DNA again. But during replication, sometimes there is an error and you insert a wrong base. Wrong base or wrong nucleotide. This is called a mutation. Now it could be just one wrong base or two wrong base or an entire length could be wrong. It is generally variable. Now this DNA is a mutated DNA. Do you think it is normal by any means? This DNA, can it be called normal? Definitely not. This DNA is not normal. It is abnormal. Now, most of the time, these errors can occur, right? The DNA is so long, you can get so many errors. But the proofreading mechanism of the DNA stops most of these errors. And even if the errors happen, what kind of genes do you think they will produce? So D is lethal genes. Definitely, if D was the answer, then survival would be really tough because if any mutation produces lethal genes, you're dead. So if all the mutation, like they generally start producing lethal genes, the organisms will not survive. Again, polygenes also, you cannot split genes that easily just by a mutation. You need evolution to occur to split genes and turn them into polygenes. Polygenes should not be the answer. Dominant genes or recessive genes. Now, again, if the gene is dominant, it will what? It will show its effect. Yeah, correct. And if it is recessive, it will not be showing its effect. Got it? So what do you think the gene is? The gene is dominant or the gene is recessive? Yeah. Mostly, the gene is recessive. Okay. So the answer is recessive genes. Now, one function of the telomere in a chromosome is to. So, you know what telomeres is? If this is a, sorry, if this is a chromosome, these end parts, which I am circling in red, these end parts are called telomere. Okay. These are called telomere. Now, if you ask me what are the function of these telomeres, let us see the options. Option number 1 says, identify the correct number of homologous pair of chromosomes. This is a very vague option. A telomere, do you think the telomere will be able to identify the number of homologous chromosomes? No, it's very unusual because telomere is not a functional unit. Help two chromatids to move towards poles. Okay. Where do you think the chromatin, where do you think the spindle fibers attach with the chromosomes? Yes, the spindle fibers attach at the centromere. This is where spindle fibers are attaching. So telomere have actually no role in that. So option number D is also incorrect. Looking at option number C, seal the ends of chromosome. This could be a valid argument. Okay. Option number D, start the RNA synthesis. Again, this is not a, we are talking about chromosomes and there is no synthesis of RNA going on in a chromosome. Chromosome, it means that the thing has already been packed up, right? Chromatin has been packed up and it is not ready for transcription right now. It is just going to go into replication, and after it is distributed and it has decondensed, only then will it be able to undergo translation, right? So that leaves us with only one option, which is the correct option. Option number C is the correct option. So option number C is the correct option. The telomere is a region of repetitive nucleotide sequences at each end of the chromosome, which acts to protect the terminal ends of chromosomes. So it seals off the terminal ends. Now, next question says, anticodon is an unpaired triplet of bases in an exposed portion of. Now, understand this. What is an codon? What is an anticodon? Okay, we'll first try to understand that. That will help us to arrive at the answer very easily. Okay. Now, codon, this word anticodon, you must have heard this a lot of times. So, there are two things. There is a mRNA, then there is a tRNA. Oh, let me try it a bit bigger. So suppose this is mRNA, and here you have your tRNA. mRNA has bases. tRNA has bases. The codon is described on the mRNA rather than on the tRNA. This is one hint. Codons in the pairs like groups of 3 are present on the mRNA. So these are codons. These codons are read by the tRNA, and tRNA brings appropriate amino acid corresponding to the particular codon. tRNA brings it, and tRNA has something called the anticodon, which reads these codons, pairs with the specific desired codon, and delivers its amino acid to the mRNA. Understood? So simple answer, anticodon is an unpaired triplet of bases in an exposed position of. So the answer will be, yeah, tRNA. We'll move on to the next question. Initiation codon of protein synthesis. This is very basic. Okay. In eukaryotes, it is always AUG, which goes for, yeah, methionine. Okay. The initiation codon for protein. Now here, okay, there is a bit of, you might be confused why they have given GUG here. Now AUG is there in, it's always, for eukaryotes, it's only AUG. And for prokaryotes, for eukaryotes, it's AUG. For prokaryotes, it can be either AUG or GUG. So prokaryotes can have 2 start codons, but eukaryotes will 100% have just 1 start codon. Got it? Okay, next question. DNA replication is, okay, conservative and discontinuous. First member of a codon second member of a codon, entire codon. Okay, these options are from some other question. Ignore these options. Okay. Okay. Option A says conservative and discontinuous. Option B says semi-conservative and semi-discontinuous. Option C says semi-conservative and discontinuous. Option D says conservative. What is this conservative, semi-conservative, continuous, discontinuous? So if this is the DNA, it has been opened up over here. This is your DNA and it has been opened up, right? And it is being replicated. Semi-conservative means only one strand is going to be replicated, okay, at a time. So in this direction, this strand is growing. The opposite direction, there will be one more growth. So because the polymerase moves in one direction, there will be at one end, there will be discontinuous growth of DNA and the other end, there will be continuous. So first, you will remove any sign of continuous, any option which says continuous. So none of the option are saying continuous. Semi-discontinuous is also we will consider. Now semi-conservative or conservative. Now conservative means the whole DNA, you take the whole DNA and you turn it into two, like that. No. But here we are using, this is called conservative. But here we are doing what? We are taking two strands of DNA and in these two strands, we are using the parent strand, one of the parent strand, and we are synthesizing one daughter strand. So both the new DNAs have one parent and one daughter strand, one of each. This is called semi-conservative method of DNA replication. Understood? So, yeah. Answer is C. Semi-conservative, in each replica, one half is the old strand and the other half is the new strand. Also, one strand is synthesized continuously and the other strand is synthesized discontinuously. Hence, it is called discontinuous synthesis. So, the answer is semi-conservative and along with that, discontinuous. Okay. All right. The next question. What base is responsible for hotspots or spontaneous point mutation? I just told you what the meaning of a mutation was. Okay. Mutation is an what? Error in the DNA. And there are different kinds of mutation. Okay. Now, hotspots. These hotspots are what? Mutations, so you know, and there are different kinds of mutations also. Okay. Point mutation, frame shift mutation. Okay. Point mutation. Just a second. Point mutation, frame shift mutation, and whatnot. Spontaneous means it is happening on its own. Now, what is a hotspot? So, hotspot is a site where a lot of these mutations are going to what happen. A lot of these mutations are going to happen, and there is increased susceptibilities of for mutation to happen. That place is known as a hotspot. Okay. So spontaneous mutation, they refer to faulty alterations in the pairing of nucleotides in the DNA. So, and in the point mutation, only single base pair is wrong. Option A says 5-bromouracil. It is a brominated form of uracil where the nucleotide is analogous to the thymine base pair of DNA. Now, instead of pairing with thymine, the adenine will pair with 5-bromouracil. This will lead the gene to cause spontaneous point mutation. So here, 5-bromouracil is going to pair with adenine. 5-methylcytosine, it is methylated form of cytosine, and it will not pair with anything. Okay. Guanine, it will pair with cytosine with triple bonds, triple hydrogen bond, and adenine makes up the DNA, pairs with thymine with double hydrogen bond. Okay. So now, as we see, the culprit is 5-bromouracil because it is binding faultily with adenine, whereas uracil is usually not present in the DNA. Okay. So 5-bromouracil is what is causing all the trouble. Got it? So the answer is option C, 5. So the hotspots was used by Benzer for sites which are more multiple than mutable than other site. Okay, the study in 1978 released that 5-methyl cytosine residues occur at the position of each hotspot. So for hotspot, they have given 5-methyl cytosine. Although 5-methyl cytosine is not a wrong option, between 5-bromouracil and 5-methyl cytosine, if they are giving only spontaneous mutation, please go with 5-bromouracil. Here, I think they have mostly focused on the hotspots. That is why they have given the answer as 5-methyl cytosine. So, okay, anyways. Next question says, DNA elements which can switch their position. So NCERT clearly mentions such elements which are switching their positions from one gene, not only gene, from one chromosome to other chromosome sometimes. And in biology, these kinds of DNA elements or genes are specially called as transposons because they are transporting themselves from one gene, one distinct place to other, and they are also known as jumping genes. Okay, so sometimes they jump from one chromosome to another chromosome also. Okay, these transposons are very active area of research right now. A lot of research is going on into these transposons, and there are a lot of implications of transposons in human genome and genomic studies. Okay, so transposons are sequences of DNA that can move around to different positions within the genome of a single cell. This process is called transposition. In the process, they can cause mutations and change the amount of DNA in the genome. Transposons are also early, called jumping genes or mobile genetic elements, discovered by Barbara McClintock early in her career. The topic went on to be Nobel Prize winning work in 1983. There are a variety of mobile genetic elements and they can be grouped based on the mechanism of transposition. So we move on to the next question. Genes that are involved in turning on or off the transcription of a set of structural genes are called. Now, set of structural genes. Whenever you hear the term structural genes and all, you should start thinking about what? Yes, operons. So, this is an operon. You will have a promoter. You will have a operator. You will have a structural gene. Now, the promoter is where the polymerase comes and binds, and it has to move forward. Now, it depends upon this operator whether this promoter region actually works or not. Because if this operator is occupied by a protein, then the polymerase cannot move forward and cannot transcribe the structural gene. So the operator is what determines the transcription of the structural genes. And this whole structure is known as an operon. Got it? So that is the function of an operator. Okay. A regulator gene is involved in controlling the expression of one or more than one genes. In prokaryotes, regulator genes often code for repressor proteins. Repressor proteins bind operators or promoters, preventing RNA polymerase from transcribing RNA. Which step of translation does not consume a high energy phosphate bond? Okay. So, which step of translation does not consume a high energy phosphate bond, which is not anything different from your ATP, right? ATP is a high energy phosphate bond. Translocation. If you remember, yeah, translocation is definitely going to need an ATP. Okay. So it will need a ATP. Amino acid activation will also need a ATP. Peptidyl transferase reaction. This is catalyzed by an enzyme called peptidyl transferase. So no ATP. Amino acid tRNA binding to active ribosomal site will also need an ATP. That brings us to the answer that yes, only option number C is one option which does not require ATP. Rest all the options will require an ATP, right? Going on to the next question. 3 codons causing chain termination are. So it is a very basic question which you must remember throughout your career in biology. UAG, UGA, and UAA are the 3 stop codons. Okay. So whenever the tRNA reads these 3 codons, the tRNA is going to stop itself, or the mRNA is going to be stopped and it will not lead to further transcription of the mRNA. So basically what it tells you is that whenever this stop codon is encountered, you need to remember that there is going to be termination of the formation of tRNA, and you should not expect that the tRNA, mRNA is going to be formed any further. Okay. So yeah. Moving to the next question. The next question asks us, which a mutation at a base of the first codon of a gene produces non-functional protein. Such a mutation is called. Now, there are different kinds of mutation. Now, they produce non-functional, functional, or semi-functional proteins. Okay. So, a mutation can produce either completely similar or a functional protein, semi-functional protein, like it performs little bit function, or non-functional protein. If it produces the functional protein as in all, it is called, it is not called, it cannot be detected as a mutation. So we don't bother about this. If it produces a semi-functional protein, then it might be a missense mutation. But if it produces a non-functional protein, if it leads to termination, then it's called a nonsense mutation. Okay. So the answer is nonsense mutation. Okay. Mutation bringing about early stoppage of polypeptide forming a non-functional protein is known as nonsense mutation. The other are due to insertion or deletion of nucleotide like frame shift and all of that. Point mutations, I've already told you, it is because of change in one nucleotide. Okay. So we'll solve the rest of the questions in the next session. Thank you.